Christos est très connu à Dauphine, et particulièrement aux politiciens de Dauphine. Comme ça?
Très bien.
Ce que je peux dire à propos de Christos Papadimitriou, c'est qu'il est parmi les 10 computer scientists qui ont façonné l'informatique depuis sa naissance comme discipline. Ces travaux à la théorie de complexité, à l'approximation polynomiale, en data-based theory, sont des travaux qui ont développé ces disciplines et ils ont créé de nouveaux problèmes à résoudre, etc. C'est le rôle d'un chercheur. Christos n'est pas... j'ai un problème. Cela ne lui pas suffit de résoudre des problèmes. Il a essayé de créer de nouveaux problèmes pour faire évoluer la discipline. Par exemple, au moment où les gens traitaient de classe P et P complet, Christos a essayé de traiter la classe un peu entière. Au moment où, à l'approximation polynomiale, on essayait de trouver des algos qui résolvent des problèmes, Christos a créé une structure pour l'approximation dans la classe P complet. Plus récemment, il a fondé la discipline de jeux combinatoires. La théorie des jeux informatiques a été créée par Christos. Et plus récemment encore, il a commencé à travailler de façon d'un théoricien sur les neurosciences computationnelles. Chaque fois qu'il fait quelque chose, il ouvre un nouveau domaine. Et ça, c'est très rare pour un chercheur. Last but not least, une discipline nouvelle comme l'informatique a besoin aussi de créer une histoire, une épistémologie pour elle-même. Christos est le seul chercheur en informatique qui a essayé de créer aussi une épistémologie de cette discipline. Ces deux livres, Turing et Logicomics, moi, je les vois comme les premiers efforts d'un informaticien de parler de sa recherche et de sa science. Au niveau de carrière, il a été prof à des plus grande université américaine. Il a fait sa thèse à Princeton et après, il a dans cet ordre, il a été prof à Harvard, MIT, École politique d'Athènes, Stanford, et San Diego, Berkeley et maintenant à Colombia. Je pense que aux États-Unis, une université, quand elle va se présenter, doit dire Christos Papadimitriou est passé ici. Et je n'ai pas d'autre chose à dire. Christos est mon directeur de thèse. Je le remercie de ça et j'espère qu'on va continuer à le recevoir à Dauphine assez souvent. Merci.
Thank you very much for the invitation. Thank you, Vangelis, for the lovely introduction. The exaggerated, typically, yeah. I'm always delighted and proud to be back at Dauphine, at my university. I know what you are all thinking. I thought this guy is a computer scientist. Why is he working on the brain? I thought computer scientists are interested in computers. And that's actually true. For the longest time, computer scientists were interested in computers, and you know, we were obsessed with computers. We worked very hard on computers. We developed all these beautiful things on computers. We were, you know, in retrospect, it's hard to believe that such a large group of intelligent adults were so obsessed with the computer, but we were. I was, okay, and we liked computers so much that we named our discipline after them, okay? So, but this lasted 6 decades, and then everything was clear. What happened in 1995? The internet came about, okay? And the internet showed us what we really want to study, okay? What is a real object of investigation? It liberated us. We felt like the mathematicians felt in the beginning of the 18th century, okay? They said, it's not circles and lines and numbers, okay? It's the universe. We are interested in the universe, okay? Every, you know, circles and equations and lines are everywhere, all right? I mean, and our mission is to study the universe. And really, that's what we did. Sort of, you know, we started studying the universe. And incidentally, I don't know if you have noticed how similar the internet and the universe look like, okay? So they're practically the same, okay? So, and when I say the universe, I mean that computer scientists have started really helping develop sciences like statistical physics, quantum physics. I mean, for example, quantum computation is in many ways a re-examination of perhaps the greatest, the most prestigious theory of the universe from the computational perspective, all right? I mean, and of course there was biology, networks, behavior, economics, and game theory. I'm going to talk a little about that. Computer scientists, so this sometimes it's called the computation as a lens of the sciences. And basically, what it means is that sometimes, in order, you know, if you know, the trick is the following, that in order to make progress in a very interesting, difficult problem, what you have to fantasize is that the object of your study, being the brain or the market or the cell, is trying to compute something, okay? And this opens up new points of view and avenues of research. And I'm not saying always, but often you have unexpected progress, okay? And I'm going to give you a couple of examples, very briefly from my past work. Nash Equilibrium was mentioned a few times today, and of course, it's very justified because, to my mind, the proof by Nash in the 1950s of his theorem saying that in every game there is always an equilibrium. In every situation where agents with different interests participate with, strategic agents in conflict participate, there is always a quiescent state. This was a watershed moment for economics. For example, it launched the new game theory, the modern era of game theory. But also it inspired Arrow and Debré to prove the fundamental results in welfare economics about price equilibria. And more to the point, to my mind, why this theorem was important. Its universality, the assurance that in every situation you have this phenomenon called Nash equilibrium, enabled economists everywhere, whenever they are studying a situation, whenever they're studying an institution they're designing or a market they're thinking about, they gave them the license to say, let's see what happens at equilibrium, right? It so happens that we proved a few years back that finding a Nash equilibrium is an intractable problem. And to me, this means that this universality is a little suspect, okay? That if the equilibrium exists, but it will never be reached within the lifetime of our universe, it makes the power of Nash's theorem a little weaker. Just to give you another example, it turns out that there is a computational way to look at evolution, and it turns out that mathematically the evolution of population of genotypes is mathematically equivalent to the situation in which the genes play a repeated game, where the strategies of the genes are the alleles, and the probabilities with which every gene plays this strategy are the frequencies of the alleles in the population. And they play a repeated game where the repetitions stand for generations. And it turns out that in every generation, the genes play through an algorithm called AdaBoost, which is one of the most successful algorithms in artificial intelligence. Okay, this came as, it was very, very near the surface of population genetics equations, but it turned out that it's very informative as to what's happening. And there is actually a very interesting dual way to look at this, that every gene in every generation, through the duality of optimization, of convex optimization, every gene in every generation turns out to optimize a trade-off. In other words, there is a trade-off in evolution, at the roots of evolution, between two interesting quantities. One is fitness, one we expect that, and the other one is entropy. In other words, the genetic diversity. So I believe that there is a lot of explanatory power in this sort of fresh view of evolution. In any event, today I'm going to tell you about the past 5 years. I've been working on this question: how do neurons and synapses and molecules, how can they produce intelligence, cognition, behavior, language, mathematics, art. Okay, this is, I hope you are interested in this problem, all right? I mean, also, okay, right. Exactly, I mean, also, when I think about this problem, I think of it as sunset from the Berkeley Hills, in the following sense, okay? Every time I watch the sunset from the Berkeley Hills, my thoughts are, why isn't everybody stopping what they're doing? And why do they come here to watch this, okay? You know, and so when I work on the brain, this is my mood, okay? Why are they working on other problems, right? I mean, it's sort of... So, and you know what everybody who works on the brain has to do? The first thing they have to do is read this book, okay? This is Kandel and Schwartz. It's a famous book, Principles of Neural Science. And every 4 or 5 years, it's reprinted, and so it's updated. It contains basically, I believe I can claim the following, that I have written a sentence from every page of this book, okay? You know, which took me a summer, okay? But as I told you, every 5 years it's reprinted, and the new edition is 200 pages longer, okay? You know, it gets thicker and thicker and thicker. Which, if you think about it, that's not right, okay? This is principles, right? I mean, and principles should shrink, right? I mean, that's not grow, right? When the Greeks set out to find the principles of mathematics, eventually they came out to 4 lines, right? Count them, you know, so, you know, this as principles is not, you know, and in fact, my colleague at Colombia and my hero in this business, Richard Axel, recently said the following: we do not have a logic, okay? I couldn't believe that he said this because sort of, you know, it describes my work so perfectly. He didn't know I was working on this. We do not have a logic for the transformation of neural activity into thought. I view discerning this logic as the most important future direction in neuroscience, okay? So, so one has to think, what could this logic be like, okay? And what I'm going to tell you today is a modest proposal, okay? You know, I think I have a good idea for this, all right? So that's, okay. So let's start at a very humble starts, okay? How fruit flies remember smells, okay? And basically what happens is that on the left, you have, you know, odorants, molecules come in to the nose of the fly. And there are about 50 receptors that catch, everyone specializes in a particular odorant. And so, in some sense, and these are projected to the neurons in the middle, the middle line, middle vertical line column of neurons, and these are called projection neurons. And in some sense, you could say that what happens is that what is formed here is a 50-dimensional vector, right? I mean, no, 52 actually, 50-dimensional vector. And then something very interesting happens. This 50-dimensional vector is projected to these 2,000 neurons called canyon neurons, and they are projected in some sort of random way. There is a random projection, so let's hope this will go away. Okay, thank you. Okay. All right. And basically, out of these 2,000 neurons, which receive various inputs, there is an inhibitory neuron here that helps select the 100 highest numbers. Okay? So for the mathematicians, what you have is a vector. You multiply it by a random matrix. You get another vector. You make the 100 highest entries of this vector 1 and the rest 0. okay? And this is how flies remember their smells, okay? So you may be not interested in this particular subject, but I'm warning you. What I'm going to argue is that this is how you are taking in what I'm saying in right now, what I'm telling you right now, okay? So, so random projection followed by cap. By cap, I mean taking the top, all right? All right, but I sort of claim that this matrix is random, all right? I mean, so what justification do I have for that? It turns out, if you can believe this, that neuroscientists, including Axel, this is the matrix, okay? They found it, okay? They dissected the fly and measured the matrix, okay? And the matrix is random, okay? They took it through all possible statistical tests, and it's really random projection, which, yeah.
You mean random, like uniformly distributed?
Uniformly distributed, right, yeah. Random, in other words, it's like as if the fly is flipping for every entry of the matrix a very biased coin, okay? And sort of this is an amazing fact, because it means that this critter with 100,000 neurons has in its DNA a random number generator. Okay, we know that it's different from fly to fly. It's different from the left to the right nostril of a fly, and so on. Okay, so it's in any event. When I say random projection and cap, that's what I know. Random projection, of course, creates a bell, and you take the upper end of the bell. And the question is, does this thing preserve similarity? If two odors were very similar, will their projections end up similar? And the answer is yes, okay? And in fact, we have a theorem that does that, that says that quantifies how similar. This is very nonlinear equation in alpha, but it turns out that for the number of, n is the number of neurons, and k, in other words, n is 2,000 and k is 100, for the numbers in the fly, this is very close to linear. If you wish, here is the line that says that it's preserved exactly, and here is what our experiments show, and this is what our lower bound is. Okay, so the point is that there is a thing called projection, all right, which includes random projection and cap, and this has functional cognitive properties, all right? I mean, which is very interesting, and it turns out that mammals have it also. Okay, so let's forget flies. Okay, does something homologous happen in mammals? And it turns out that the olfactory system that we have is very similar to that. Okay, we have 500 smells, odors instead of 50. Dogs have 2,000. Okay, and we lost 3/4 of them on the way. But basically, we do it exactly the same way. We get odorants. There are olfactory receptors in our nostril. The olfactory receptors, when they are excited, they excite the olfactory bulb, the particular glomerulus, the particular part of the olfactory bulb that is responsible for this particular smell, like tarragon. And then there is a projection to an ancient part of our brain called the piriform cortex. And this projection has the properties that I told you before for the fly, except it's much more complicated. It's complicated in a mammalian way that was not available in the fly. In particular, let me show you how Axel and his co-authors describe this. An odorant may cause a small subset of neurons to fire. Inhibition triggered by this activity will prevent further firing. These first two lines are the fly. Okay, there is inhibition, and then there is random projection. This small fraction of cells will then generate sufficient recurrent excitation. Here is the secret, that in mammals, the piriform cortex has recurrent connections. In other words, there are synapses from neurons from one neuron of the piriform cortex to the other, whereas this was not true in the fly's brain. Okay, there was only the bipartite projection that I showed you. And in the extreme, some cells would receive enough recurrent input to fire without receiving initial input. Let me show this to you okay, the animations are not working. I apologize. So let me tell you what happens. This fires. Imagine that this is pulsating, and it creates this what it would create in the fly's brain. Now both of these will continue firing, both the blue and the black, and will create a new set of winners. Okay. Then the red and the black will fire, and they create a new set of winners, and eventually, hopefully, this will converge, and that will be the stable set of neurons that are going to represent the particular smell, okay? And that's the moment, actually, that you will say, oh, I like tarragon, okay? You give it a name, right? All right. So, and also something that I call Hebbian plasticity. What's Hebbian plasticity? Plasticity is how brains learn. Hebbian plasticity is a very simple rule, that if a neuron has fired, and as a result another neuron fired, then this is a useful connection and should be increased in strength. Okay, this is what Hebbian plasticity means. Yeah, of course. Okay, good. Does this process converge, and does it preserve similarity? And let me tell you, before answering that, sort of, you know, I'm not going to show you the mathematical theorem, but I want to show you the model, because believe it or not, we have a model of the brain. Okay, so you know, when was the last time that somebody told you this? We don't have a model. You know, we don't care for you know, to what 95% of neuroscientists care for. In other words, modeling the visual cortex or the auditory cortex, or, you know, we're not interested in the senses. We're interested in what is called the associative brain. We're interested in the inner life of the brain, okay? Much of neuroscience is concerned with what neuroscientists can do. In other words, experiments where they show to animals things and they watch what they do and how their brain reacts, okay? So, you know, so I believe that this is a well-studied part of the brain, and it's in good hands. But I'm more interested, first of all, in human intelligence, and second, in the functions that I've told you before. I mean, how do we do language, for example, okay? And I believe that this is an interesting way of looking at the brain, that it's a finite number of brain regions. I mean, think of them as 1,000 brain regions, perhaps. Each one has, I don't know, maybe 10 million excitatory neurons. Without, you know, so imagine that each one of them contains 10 neurons, and inhibition means that only K of these fire. It's exactly the random projection in cap once again. Some pairs of areas are connected by directed arrows, by directed connections, and these connections are random, as in the fly. All are connected recurrently. In other words, within every one of these regions, there are recurrent connections that are again random, okay? And basically, neurons fire in discrete steps. This is an assumption that are very useful, very important for what we do, and it's as useful as it is indefensible, but I don't think that it changes the mathematical situation very much. Connections between areas can be enabled and disabled, and there is plasticity. If there is a synapse from I to J, I fires, in the next step J fires, then the weight of I to J is multiplied by some number greater than 1. And then there is also homeostasis for getting things that I'm not going to tell you much about, but sort of, you know, they make this a reasonably convincing model of the brain. The main idea, there are three. You know, what I'm saying is that the part of the brain that I'm interested in, I think there are three main physical forces that work. One is randomness. In other words, the randomness of connections between neurons is very important. Selection through inhibition. The highest winners are selected. There is a mechanism for doing this very nonlinear operation of selecting the biggest numbers. And plasticity, okay, the change in the weights of connections. And our theorem says that the projection process that I told you converges with high probability and the total number of cells. So we have a reasonably accurate analysis of what's happening. Okay, and these are the results. When you have large plasticity, things converge extremely fast. When you have small plasticity, things again converge. All right, and this is a typical pattern. After a dozen steps, you have a very stable assembly, as we call it. The result of such projection, let's call it an assembly. It's a set of about k neurons in the brain area, firing in a pattern whose firing is tantamount to our thinking of a particular memory, concept, name, word, episode, and so on. Okay, so it's a unit of memory, if you wish. That's not my idea. That's not a new idea. That's old history. Okay, you know, this is something that 70 years ago Hebb conjectured. And it has been sought and found, discovered many times, and then disputed. And finally, through very modern technology, people actually found that, yes, these things exist in our brain, okay? And basically, we know that, through simulations, we have a very apt simulation system. And what I'm telling you, what I told you, what I'm going to tell you, we know that this happened at least in our model. All right. You remember, there was this myth of the grandmother cell, okay? That there is a cell in our body, sort of, that in our brain that where our grandmother resides, okay? That can't be accurate, okay? If it's one cell, if there is one grandmother cell, then there is no grandmother, right? Because the effect of one cell cannot be significant enough for us to bring the image of our grandmother to our brain, okay? So it's probably 40,000 cells, right? Nowadays they're not called grandmother cells. They are called Jennifer Aniston cells, okay? And the reason is that they found them, okay? They found in the 90s, during the 90s, they recorded from people, they showed them Jennifer Aniston, and this cell consistently fired when they saw the image of Jennifer Aniston, or even her voice, or even her name printed, okay? And of course, also Travolta, okay? And all that. The numbers tell you that if they showed 50 pictures and they were able to find the Jennifer Aniston cell, probably there are not one Jennifer Aniston cell, but maybe 60,000 Jennifer Aniston cells in the same brain area, right? So that's the idea. So these are called Jennifer Aniston cells or concept cells. By the way, after my talk, you are going to have not just Jennifer Aniston cells. You are going to have Jennifer Aniston cells, okay? Think about it, okay? But don't go this way, okay? It's not. All right. So in any event, let's talk about the big picture, okay? So my ambition, computation in the brain, okay? What is that? By the way, people ask me, Professor, do you really think that there's computation in the brain, okay? Frankly, I don't think that's a scientific statement, okay? It's a methodological statement. If I'm helped in my research by thinking that there is computation in the brain, then there is computation in the brain, okay? This is, all right. So what is the right level? Molecules compute, of course. Spiky neurons and synapses, of course they compute. People think that the real computation happens in the dendrites, in the input devices of the cells, okay? And cognitive scientists, they have this whole brain computation, where basically they make an experiment, a cognitive experiment, and they say, see, the brain of the subject is as if it's executing this program, okay? So that's called whole brain computation. It's very useful in cognitive science. The question is there something in between, okay? Is there something like the logic that Richard Axel wants to see, okay? remember his invocation of this particular mysterious logic, okay? So here is my hypothesis, the assembly hypothesis. There is an intermediate level of brain computation. It's implicated in carrying out several higher cognitive functions, such as reasoning, planning, language, storytelling, math, music, and so on. Assemblies are its basic representation, its main data structure. And the question is, what are its fundamental operations? And by the way, an operation must be useful and plausible. Useful means must help explain experiments. In other words, it must explain something that people, neuroscientists, have found in humans or animals. Should be plausible, can be compiled down to neurons and synapses. In other words, we should have a way to explain these operations through individual neurons and synapses firing. Okay, you know project is an operation. It's exactly what I told you. That's how assemblies are created, and that's how they can be copied further for further processing in other brain areas. Okay, other operations. Two assemblies may be associated by sharing cells, okay? So if two assemblies represent two things, like your Vangelis assembly and the Papadimitriou assembly, from now on, they're going to have some overlap, okay? You know, that's what I'm saying. I mean, they should overlap because these two concepts have some affinity, similarity, co-occurrence, and so on, right? So, for example, I mean, the idea is that if these assemblies, okay, I jumped this because, as I told you, my animations are not working. Okay, so that's going to be a problem then. Here is the Aniston. Here is an amazing experiment. Okay, so I'll try to walk you through it without the animations. So they recorded, let's say, they recorded from dozens of cells, from dozens of humans, but let's focus on one human and one cell, okay? And this was the Eiffel Tower cell, okay? That every time they showed an image of Tour Eiffel, this neuron went wild, okay? They showed them other things, sort of, you know, nothing. They showed them, every time they showed them the Eiffel Tower, it went. They showed him people they should know, nothing, okay? And then they did something clever. They photoshopped Obama in front of the Eiffel Tower, okay? You know, so they probably did it more artfully than I did, okay? You know, but the point is that the cell saw the Eiffel Tower and dutifully it fired, right? Then they showed the Eiffel Tower, of course, it kept firing. They showed the Forbidden City, nothing. And they showed Obama and fired. You see it? So, I mean, I cannot say this with dry eyes, okay? You know, this guy learned that Obama has been to the Eiffel Tower. And you tested this experimentally. You know, you found this experimentally. This is amazing, okay? You know, basically what happened is that there was an Obama assembly which they didn't touch. There was an Eiffel assembly which they found. And some of the cells in the Eiffel assembly migrated to the Obama assembly, okay, because the person found that Obama and the Eiffel Tower were related, okay? So this is not just a single experiment. It has been repeated many times, and you know much more now about such associations. Okay, so, and in fact, association of assemblies seems to be very well preserved under projection, okay? You know, so there is association and there is projection. Other operations, okay? So, you know, I don't want to go over time. There is something that we call merge, okay? And that's important. We have a project, we have association, there is a third operation. There are more, but so, you know, let's concentrate on these three, they're enough. Merge, and merge basically means that two assemblies create one thing that is basically a tree that has two leaves and creates, you know, it's a hierarchy, an elementary hierarchy. And of course, this can continue on and on until you create many hierarchies. And that's what, it's valuable for implementing language. And it's complicated. To do it, we know how to do it, we simulate it, it happens, except that it requires a very careful, very sophisticated coordination between 5 brain areas and that are connected in a very strong way. And the question that's in my mind is, does it need enhanced hardware? I think I'm going to jump to the end of my talk, and I want to finish with this. I want to emphasize this. There is something in our brain called the arcuate fasciculus, okay, which joins this part of our brain to this part of the brain. This is called Wernicke's area, and this is where we stored words, okay? And notice I'm working on my left hemisphere. This is Brocas's area, sort of French doctor, and this is where syntax happens, where sort of, sentences and phrases get compiled. And this thing joins these 2 areas, okay? It becomes operational near the 2nd year of the baby's life. So it turns out that on the left side, this thing is 60% to twice as big as on the right side, okay? And it's no other mammal has such asymmetry, okay? That this is the only case of left-right asymmetry, I mean, physiological asymmetry, anatomical asymmetry in the mammalian brain. And it happens in the language, okay? So there are clues here, I think, all right? I mean, in any event. The assembly operations, these are the assembly operations that I talked about. It turns out, if you ask how powerful is the system, there is a simple answer, there is a sort of a very interesting answer. It can perform, it's Turing complete. It can perform full-blown computations, okay? And I mean, usually when I read a paper in biology that shows that something is Turing complete, I stop reading, okay? But this is a system that I am hypothesizing can do things like deduction, okay, and language. So I need to show this, okay? All right? In any event, language. This image was supposed to show you that my colleagues, my neuroscientist colleagues, try to dissuade me from working on language. Okay, they tell me, why don't you leave this for last? Once we understand the brain, we are going to lie back, light a cigar, and think about language, okay? But I think that language is an opportunity. That language is something that evolved after our brain had stopped evolving, okay? So if we want to understand how the brain works, how our brain works, we should study language, all right? I mean, so, okay. As a bonus, I'm going to show you sort of a beautiful experiment by a cognitive scientist at NYU called David Peppel. So here's what he did. It's very hard to, it's sort of, you are missing 90% of the fun without the animations. But what he did is he read to patients, patients heard the tape with words at 4 hertz, something like Fred Ship, he'll give true melt fans blue, guess hits then cuts, okay? And in the end, he recorded, and then he took the Fourier transform of the recording, and of course he found a peak at 4 hertz, because 4 times every second, the brain has to fetch this word and understand its meaning and groping if there is any connection between words, all right? I mean, so that's 4 hertz is easy to explain. Now he repeated the same, except that every 4 words make sense, okay? They create a sentence. And it turns out that there is of course a 4 hertz peak, but then there is a 1 hertz peak, because it means that every second, this person created a sentence. And there is a 2 hertz peak, because twice a second, this person created a phrase, okay? And if you ask me what happens, I think that this is happening, okay? That basically these people are building trees, okay? They're building trees in their brain. And we have a, you know, I'm going fast over this. We have a conjecture of how this happens, and it's consistent with a couple of dozen of experiments that we know. Okay, we have a conjecture of that it's a bunch of projections, a bunch of associations, and a bunch of mergers that create the trees that again is our interpretation of what happens. All right, so I'm rushing you through, but this is my conclusion. I don't know if you know the story, but 130 years ago was an amazing moment in neuroscience. In fact, it was the birth of neuroscience, because two great scientists in Europe, one Italian, one Spanish, they saw this picture, okay? The first one, Golgi created this picture, right? And what Golgi saw, okay, he saw the whole picture as one, and he said, the brain is a network. We should understand how networks work, okay? I mean, also, he was the reticulus theory, right? I mean, also, whereas Ramon y Cajal, Santiago Ramon y Cajal, saw the same picture, and he noticed this, okay? He noticed the neuron. He said, this is a neuron, folks. We should spend 100 years studying the neuron, okay? The question is who was right and who was wrong. And of course, they were both right, and of course, they were both wrong in being dogmatic and rejecting. You know, they had, you know, they both got the Nobel Prize, but they had an epic fight in Stockholm, okay? So they were both wrong in rejecting the other point of view. But in some sense, Ramon y Cajal was right, because there was thousands of physicians who were ready to study the neuron, okay? Whereas there were not thousands of scientists who were ready to study the network in 1890, okay? So in any event, I guess what I'm telling you is that there is another, you know, maybe there is another way. You look at this picture and you see sort of, you know, an assembly of neurons. And this is the unit that is worth building a theory on. In conclusion, there is no end to the study of the brain, and there is no end to the fascination that it provides. I believe that assemblies and their operations will be one productive path to thinking about it. Could they be what Axel is looking for? An important thing, you know, we believe we understand how assemblies can do syntax in language. We don't know how they learn and how they predict, okay? This is how they can be sort of, you know, the data structure of efficient learning algorithms. And ultimately, how can one test, verify, or falsify the assembly hypothesis through experiments in neuroscience? So these are the people I work with: Wolfgang Mass in Austria, Santosh Vembala in Georgia Tech, Larry Abbott is the leader of the theory group at neuroscience at Colombia, and my colleague and collaborator with this. Dan Mitroposki is my student, and Mike Collins is a linguist at Columbia. So thank you very much.
Est-ce qu'il y a des questions?
I'm judging by two things. The first, you don't speak so much about the plasticity. I would like more about it.
Sure. Yes, yes.
And the second point for Christos, in fact, the dynamics of brain is something. And you don't speak about how to learn in the learning process. For instance, the computing of a novice, for instance, in board game.
Of computing?
Suppose a board game. And you look at the difference between the brain of a novice, and the brain of an expert. It doesn't work in the same way, because there are many
Definitely, yes. Thanks for your questions. Plasticity is, as I said, is how brains learn. The way we model plasticity is particularly simple. We now, since the 90s, we understand a lot about plasticity, okay? We have a very simple model of plasticity because we want to keep our mathematical model as simple as possible. So the model of plasticity is Hebbian plasticity, that if two neurons fire together and are connected, then this connection is rewarded by an extra factor, okay? So this is, now we know so much about plasticity that it's, it's almost too much, but I believe that this is, and neuroscientists I talk to also believe that it's a good start to use Hebbian plasticity in a very simple model of what we want to do. Then you ask me, what's the difference between an expert and a novice chess player? Okay, so sorry. The plain answer is I don't know. I'm pretty sure that chess or Go configurations they are essentially episodic memories, and they are represented somehow, possibly through assemblies. The question is what circuitry is built over the years with sort of coming again and again in contact with the same subject, with the subject of playing Go, for example. And as I said, I am very interested to find the ways in which these representations can do learning. And I'm going to start with much less demanding learning than learning a particular board game. I'm interested in how we learn to distinguish between circles and squares. Okay, this is in other words linear separation. Okay, how assemblies can possibly do linear separation. I mean, one thing that is encouraging is for those in the audience interested in learning theory, is that this random projection and cap operator that is the basis of what I'm doing is, in some sense, it has very interesting analytical properties. It's a very simple but analytically very complex function, and it is it's seen as a kernel in learning theory. It is a very apt kernel. So I have hope that it can enhance through linear learning. It can do several simple steps in linear learning.
Thank you, Christos. Very interesting lecture. So one of the things I learned recently is apparently we can get much better at smelling, which is the learning question we can get much better at smelling. You said we lost a lot of our receptors through evolution, whatever, but apparently we can get them back or whatever. Some people can get trained to smell much better. So that will fit into your learning thing, right?
Yes.
So you think that what happens there is that you use the same receptors. So the ones we lost, but now we're using the same receptors more efficiently.
Oh yeah. We cannot smell female dog at all. Okay.
Okay.
We have lost that forever.
We have lost that forever. So what is it that we learn how to use more efficiently the same?
Okay. I mean, I gave you only a small part of the picture.
No, I understand.
Okay, right? I mean, so there are, after the smell goes here in the piriform cortex, right. It is projected in another big area here, and there very interesting things happen. Okay, so people only now. Axel got the Nobel Prize for olfaction because for the olfactory receptors, but he has gone on. I mean, so now he's working on this. I mean, how this projection from the piriform cortex to more areas that are much more apt for learning happens. And probably these are the processes that can be enhanced through training, through selective training. So, they're writing, there are many recent papers about it. I'm not familiar with all of them, but there are many recent papers coming out from my group, at Colombia, but I'm not very familiar with them.
Thank you.
Sure.
Any other question? Yeah, at the back.
Non-determinism. So I'm not even sure that statistical analysis is the right. Statistical analysis will be used, of course, in experiments a lot, but in understanding experiments. But my vision is that you can start understanding. Okay, what happens in our brain? I don't think I'm ready to start imagining. Okay, so what I showed you is a cartoon. Okay, so these assemblies are a cartoon. The real story is going to be much more complicated, much more dirty, much more wet, much more organic. Okay. And messy, but my hope is that this is a mathematical model that can summarize the story adequately well. Okay, so my plan is to continue with this kind of mathematics, probabilistic analysis, combinatorics and probabilistic analysis, to create this cartoon. Okay, to finalize a great bigger picture of this cartoon, and experimental neuroscience will come up with better and better recordings of what really happens, and we'll see how they jive with what we find.
Rida.
Christos, so here you try to use computer science to understand brain. Can the opposites also be useful? Can you imagine that from the brain you can discover some algorithms and architecture that may be very useful in computing?
Okay, that's okay. I'll tell you something that please don't repeat. Okay, so when I write research proposals, I write a page where I say, I hope that this will happen, okay? When I look inside me, do I ever hope that this will happen? I'm very pessimistic, okay? I think that the brain works in ways that it has inherited from the fly, from the mouse, okay? So it's trying to do amazing things with its hands tied behind its back, okay? So I don't think that that will find something that we can duplicate. I'm ready to be surprised, okay? But I'm not expecting that representing a bit by 60,000 neurons will be a clever idea ever, okay? So the brain is doing it because it inherited the trick from the fly, I think.
Okay, then thank you again for this very nice talk. Et je ne vais pas conclure par un long discours, je vais juste remercier encore une fois tous les orateurs qui seront venus de loin ou de moins loin nous faire la gentillesse de participer à cette journée. Merci encore.